Hi, my name is Jesse Hopkins, and I'm a staff scientist at the BioCAT Beamline, which is Sector 18 at the Advanced Photon Source. So what I'm going to talk about today is analyzing flexible and disordered systems using small-angle X-ray scattering. So when I talk about SAXS and flexibility, I want to first define what I mean by flexible systems. Flexible systems adopt a continuous range of conformations, at least for the purpose of this talk. And I want to make this distinct from polydispersed systems that adopt a small number of kind of distinct states. Just a couple different conformations, a couple different oligomers. And this is because the analysis for these different types of systems proceeds differently for SAXS data. Additionally, I'm going to use the terms flexibility and disorder kind of interchangeably in this talk. You can argue if they're the same or not, but flexible regions tend to be disordered in a protein, disordered regions in a protein tend to be flexible. So it turns out that the measured scattering profile is a combination of all the different species and conformations in solution. In particular, it's this volume-weighted sum of all the individual scattering profiles. This can sometimes be painful for SAXS because you can measure a system with some aggregate in it and you get a scattering profile that comes from both the aggregate and the sample of interest, but for flexible systems it's an advantage. So it means the single SAXS measurement samples the entire conformational ensemble. And in particular, this makes SAXS one of the few methods that can quantitatively characterize partially disordered or completely disordered macromolecules. And this is often used in combination with NMR for this purpose. So the first question we really need to be able to answer when we're thinking about flexibility in SAXS is my system flexible, right? Before you can proceed with any of the kind of fun advanced analysis, you need to be able to tell if you're measuring a flexible system. And the way to do this is using multiple different metrics from the SAXS experiment. And this is because any individual point that I'm going to go over here could be caused by either flexibility or something else, as I'll tell you in a few slides. So characteristics of the scattering profile: it's generally smooth, there's a plateau or an increase in the dimensionless Kratky plot, there's no plateau in the Porod-Debye plot, and the Porod exponent can be less than 4. There's some characteristics of the P of R function. It tends to be smooth with little or no fine structure. There's an extended tail usually. Dmax can be hard to determine. And RG and I of 0 from the P of R function are usually larger than from the Guinea fit. We also get overestimates of the molecular weight from volumetric methods, and there's a few other things which I'll mention in a few slides. And so what I'm going to do is I'm just going to go through kind of each of these point by point and tell you why they happen and why they indicate flexibility. So first, we're going to start with the scattering profile. And so for fully disordered systems, the scattering profile, I of Q, is characteristically smooth because it represents an average of a large number of conformations which washes out distinct features. And so I think this is really nicely shown in this figure here on the left. So this is a group that collected data on an intrinsically disordered protein, an IDP. That's these gray circles. And what they did to analyze it is they simulated 5,000 different possible conformations and generated the scattering profiles for each of those conformations. 7 of those conformations are shown here on the left, and their corresponding scattering profiles are shown in the colors in these two plots. And what you'll see, especially emphasized in plot C, is that each of these individual profiles that correspond to one conformation have distinct features, right? There's dips, there's rises, and they each have some kind of interesting shape. However, when you average all 5,000 different profiles together, all these individual shapes of the profile kind of average out into this single, extremely smooth, uniform curve shown here in black. And so this is what we mean when we say that the scattering profile is characteristically smooth. You can also see that in this data over here. So this is simulated scattering profiles for a globular, 50% disordered, and completely disordered system. And you can see as we increase the amount of disorder in the system, we get fewer and fewer kind of distinct features as characterized by these minimum and maximum, these local dips and rises. So that's one thing you can look for. The next thing I want to talk about is the Porod exponent. So Porod's law, which is also known as the Porod-Debye law, states that at high Q, the scattering intensity decays as Q to the negative D, where D is the Porod exponent, and it depends on the particle shape and flexibility. So there are some cases where we can actually analytically calculate out the Porod exponent for particular kind of model systems. And so these known Porod exponents: hard spheres are 4, rods are 3, thin disks are 2, Random walks, as modeled by Gaussian chain, is 2. This is kind of representative of natively disordered proteins. Extended unfolded proteins, as modeled by self-avoiding random walks or swollen Gaussian chains, are about 1.7. And then needles, so very thin rods, or fully extended chains, are 1. And so you can see as we kind of go down this list, as we get smaller and smaller exponents, we're getting less globular systems, which indicates either flexibility in the case of these kind of random walks or self-avoiding random walks, or anisotropy in the case of these rods or discs or needles. And so if you have a Porod exponent that's lower than 4, it can be indicative of flexibility. Now, Porod's law actually breaks down at higher Q due to either shape effects coming from folded or partially folded systems, or hydration effects and excluded volume effects, which will hit all systems. And so to get an accurate Porod exponent, what you really want to do is you kind of want to fit in the mid-Q range. Scatter is a really nice way to do this, and you can get the Porod exponent from there. And so in this plot on the right, I'm just showing you this Porod exponent region. So this is a log-log plot, which means that a power law, like q to the negative d, looks like a line, and the slope of that line is the exponent. And so for this folded system here, you can see there's a linear range somewhere in here with a relatively steep slope. For the unfolded system, there's this long linear range, and it has a shallower slope. So this corresponds to a smaller Porod exponent, closer to 2 probably for the unfolded system. The folded one is larger, closer to 4. This is what we expect. And we actually use kind of this existence of the Porod region where it decays like this to our advantage in several ways. So the first of these is with the dimensionless Kratky plot. So a normal Kratky plot is q squared times I of q versus q. But this can be somewhat problematic because it's hard to compare between different sizes and concentrations of protein, for example. And so what we tend to do is we tend to put the scattering profiles on a dimensionless scale. And this removes the effect of size and concentration and allows direct comparison of shape and conformation in the system. And so we do this by multiplying every q by an Rg and dividing I of q by I of 0. So we end up with a dimensionless Kratky plot, which is qRg squared times I of q over I of 0 versus qRg. And this is taking out those kind of shape and concentration effects from our system, and overall size of the macromolecule as well. Sorry, it's not taking out shape, it's taking out size and concentration effects. So it turns out that globular systems have a very characteristic shape. They tend to have this very kind of Gaussian bell-like shape here, shown here, and they have a peak of 1.1 at square root of 3, which is about 1.73. Random chains, so natively unfolded systems, come up and have a plateau somewhere between 1.5 and 2, and then at high Q, they might increase further due to hydration effects. And fully extended chains just continue increasing beyond 2. And so you can see kind of some examples here on the left. So you have a globular system that comes up and comes back down, very characteristically. You have the natively unfolded system that comes up and has a plateau. And then you have this 50% disordered system, which is very characteristic of systems with some flexibility, like several domains connected by a flexible linker, where it comes up, it has some kind of plateau, and then it starts coming back down. You can also see that shown very nicely here in this figure on the right. So here they have real scattering data for different systems. So this purple is a very extended chain, so the Porod exponent is less than 2, which means when you have this q squared times I of q, it increases as some power of q, and so it keeps just increasing out here at high q. So this gray curve is essentially randomly disordered with maybe minimal secondary structure. So this has a Porod exponent close to 2, which means that when you multiply by q squared, you get a constant, so it comes up and it levels out and has a plateau. These flexible systems in red and green, one is kind of more extended, more extended flexible linkers, the other is more compact. You can see they both come up, they both have some kind of plateau, they come back down, but they do so at different points. And again, thinking about the Porod exponent, right, it's probably somewhere between 2 and 4. And so you get, instead of a plateau, you get a more shallow decrease than you do for a globular system, and it comes out to higher Q Rg here. And then finally, you have this very compact globular system. It comes up, it has a peak right where we expect it, and then it comes back down. And so this is the kind of thing we expect to see on a dimensionless Kratky plot. And you can use this to help interpret the kind of shape and flexibility of the system. So in addition to dimensionless Kratky plots, we also use what's called the Porod-Debye plot. So this is q to the 4 times I of q versus q to the 4. And so on this plot, instead of looking at the entire plot, we tend to look at just kind of the low to mid-q region, concentrated at just after the first peak of the Kratky plot, really, if the Kratky plot has a peak. And so this plot can be useful to help distinguish between globular and flexible systems when the Kratky plot is indeterminate. And in particular, it's useful when looking at systems to try to determine between complete flexibility or a flexible and a globular domain. You'll also sometimes see the use of q to the 3 times I of q versus q to the 3 plots. These are sometimes called Sybil's plots, and that can also help. So essentially what we're looking for on this plot is, again, thinking about the Porod exponent. If you have a globular domain, at some point, it's at least been relatively well shown by this paper here, there's going to be a region that's decaying as q to the minus 4. And so on this plot, when you have q to the minus 4 times I of q, that means there's going to be some region with a plateau. And so a plateau in this plot indicates a compact globular domain with minimal conformational flexibility, whereas the lack of a plateau indicates either full flexibility or an extended object, a non-globular domain. And so this is shown very nicely with this sample data here. So here they were looking at an intrinsically disordered protein, an IDP, shown in red, and the same system but with an MBP tag, which is compact and globular. And so you can see very clearly the differences in the Kratky plot. And then if you look at this Porod-Debye plot, you see the black curve, which is the curve with the MBP tag, comes up and has a Porod plateau. So this is what we call this leveling out on the Q4 I of Q versus Q4 plot. Whereas the intrinsically disordered system, which has no compact globular rigid system, just continues going up. It has no plateau. And so this plateau here is indicative of some kind of compact globular system with minimal flexibility. So in this case, it's fairly obvious even from the Kratky plots that they're very different, but there's a nice illustration here on the right where you're looking at Kratky plots in this inset here that are fairly similar. And so what they're looking at in particular, it's this protein with ATP bound and unbound. And so when it binds on the Porod-Debye plot, you see it comes up and there's a plateau. And they attribute this, as we've said, to this kind of compact globular domain with no flexibility. When it unbinds, what you're seeing is there's no lack of plateau. There's no plateau on this plot. And they're attributing this to there being significant conformational flexibility without ATP bound. And so in this way, it can help kind of distinguish what the Kratky plot is actually telling you. So that's what we see when we look at the scattering profile. Now I want to talk about the P of R function. So the P of R function for fully disordered systems is characteristically smooth, very similar to the scattering profile. Because again, it represents an average of a large number of possible conformations, which washes out distinct features. And that's shown very nicely in this illustration down here on the left. So this is the same IDP data I showed a few slides back. You can see again the simulated conformers, and you can see their calculated P of R functions this time. So you can see each of these has kind of distinct shapes. The purple in particular has that double peak. The orangish one has a shoulder. But when you average all 5,000 together, you get this single black scattering profile here. And so this is very characteristic of it being flexible, is this very smooth, featureless P of R function. The one feature we tend to see in a flexible P of R function is an extended tail that gives a slow approach to Dmax. Additionally, you might find that the maximum dimension is hard to determine because you have some small number of conformations sampling extended sizes, and so you get very few, but it kind of drags out the P of R function for finding the maximum dimension on the molecule. Also, very characteristically, Rg and I of 0 for the P of R function are larger than from the Guinea, and they tend to be more reliable. So you can see the extended tail in this simulated data here very nicely. So we have P of R for a simulated globular system in dark blue, for the 50% unfolded system in light blue, and for the completely unfolded system in gray. And you can see as we add in disorder, we get this longer and longer tail on the P of R function. This is also, I think, kind of nicely illustrated, the smoothness anyways, in this example here. So this is P of R functions for a system where you have globular domains connected by a flexible linker. And so in green you can see that case measured with a flexible linker, it's relatively smooth, whereas in black they made a mutation that made the mutant is extended and relatively rigid, this linker. And you can see there you have a lot more shape to the P of R function. So it's a nice illustration of kind of the smoothness that we expect. So in addition to these effects on the scattering profile in the P of R function, there's also an effect on the calculated molecular weight. So flexible systems in solution occupy a larger volume as measured by SAXS. Each individual conformation, of course, occupies the volume of the protein, but SAXS, you have to remember, it measures all the possible conformations in solution, and so to our probe, at least, it looks like this protein, when it's flexible, is occupying a larger volume, right, the space of where all the conformations is, rather than just the space of a single conformation. And this means that molecular weight calculations that depend on volumes will tend to overestimate the molecular weight for flexible systems. So this includes Porod volume methods and the molecular weight estimated from 3D reconstructions. So another way to approach this is if you have a molecular weight and oligomeric state of the sample and they're known, you can actually calculate the observed particle density from the Porod volume. For proteins, we expect a particle density of about 1.36 gram per centimeter cubed. Globular proteins in SAXS tend to be between about 1.3 and 1.4. Flexible proteins tend to be between about 0.9 and 1.3. And so you can use these molecular weight differences in these volumetric methods versus other methods, or a low calculated density as indicators of flexibility. There are a few other indicators of flexibility that I just want to mention. So first off, the Guinea fit may only extend to Qmax times Rg of about 0.8. This is particularly for intrinsically disordered proteins. If you're calculating the Fleury exponent, which defines the relation between molecular size and the number of residues, and is 1 over the Porod exponent, you're going to get, usually for IDPs, somewhere in the range of 0.5, 0.6, globular systems 0.3, or something else. This is a fit assuming specific models, so it can only be done if you have a specific model for the system. And then if you get all the way to doing reconstructions or rigid body modelings, assuming one shape, the results tend to be characteristically extended because they're trying to fit these extended flexible conformations in solution. And the reconstructions tend to show large volumes for flexible regions. This is indicated nicely in this set of figures here. So this is simulated data for this two-domain complex with either a flexible linker or a rigid linker. And you can calculate the bead model reconstruction shown in blue. You see that for the flexible linker, it's occupying kind of all the space in this flexible linker region. And there's not really a distinction between the two domains. Whereas in the rigid case, it very clearly follows the linker quite closely, and you can easily distinguish the two domains. So this is what you might see with a flexible reconstruction. I do want to point out that a high normalized spatial discrepancy in a reconstruction is not necessarily a sign of flexibility, nor do flexible systems necessarily have high NSDs, and this paper here goes into some of the details nicely, so you can check that out for more information. So now I've gone through a bunch of different indicators, and the question is still, right, is my system flexible? And the answer is, well, how many of these different indicators are saying it's flexible? Because each of these indicators of flexibility can be caused by something else. So the smoothness of the I of Q function, the scattering profile, or the P of R function can be caused just by a particular protein shape. Particularly for globular systems, the P of R function is extremely smooth. The Porod exponent being less than 4, or the plateau or increase in the dimensionless Kratky plot, or the lack of plateau in a Porod-Debye plot, comes out of the particle shape potential instead of flexibility, right, just being more anisotropic. Additionally, the dimensionless Kratky plot needs good background subtraction, so if you have bad background subtraction, that might look like flexibility. An extended tail on the P of R function, or Dmax, that's hard to determine could be due to aggregation in your system instead of flexibility. Rg and I of 0 from the P of R function that's larger than the GAJOE could just be from a poorly determined maximum dimension. It tends to increase Rg and I of 0. Overestimates the molecular weight. Well, the simplest option is that, for example, maybe you think you have a monomer, but it's actually a dimer in solution, so it looks overestimated. Or maybe there's an aggregate that's causing that, or some of the inherent uncertainty in the molecular weight methods. Narrow GAJOE range could come from trying to overfit aggregation or repulsion in your system. Having a lower Flory exponent could just be from poor fitting due to a bad choice of the model. And extended reconstructions of rigid body models could just be coming from poor reconstructions with high ambiguity or an actual extended particle shape. And so because of this, because you can get each of these indicators from something else, you really have to combine several different indications to make a statement about flexibility. Really, determining flexibility from SAXS requires a preponderance of evidence. And you need to be able to eliminate other possibilities. So just as kind of an example, suppose you make a measurement. The first thing we usually do is a GAJOE fit. If the GAJOE fit looks good, you can, eliminate at least significant amounts of aggregation or repulsion in your system. So now you do a normalized Kratky plot. Suppose that's coming up and leveling off. And you want to know, is that due to flexibility, or is that due to, say, it being kind of disk-like and having this anisotropic shape? So then you can create the P of R function. You can compare the shape of that to the shape of, say, a disk or a rod in solution. If it has the characteristic extended tail, if it doesn't look like one of these anisotropic geometric shapes, then combining that with the Kratky plot, combining that with the GAJOE to discount aggregation, so you know you're not getting that extended tail from aggregation, and additionally other indicators like maybe the molecular weight, then you can say with significant confidence that your system is likely flexible. And so it's combining these multiple metrics that becomes really key for defining flexibility in SAXS and saying whether or not you have it. All right, so once you think you have flexibility, once you know you have flexibility, you want to do some kind of advanced analysis to learn more about your system. So flexible systems did not exist in a single conformation solution. That was kind of the definition that I started with. And so the best that we can do with analysis is we can either address kind of what range of conformations are in solution, what they might look like, or in some cases, what a single average conformation might look like. And so typically what we do is we use ensemble methods for analysis. These describe a set of possible conformations and then the set of actual conformations in solution. You can potentially use single conformation methods to describe an average conformation, but these should only be used for tightly peaked ensembles. And so there's some papers here that I've cited, actually, two papers, which go through kind of some of the applications and limitations of these single conformation methods, like a bead model reconstruction or rigid body modeling. But just to emphasize, these can't describe the full range of conformations. So you really have to proceed with caution on tightly peaked ensembles or proceed not at all. What I want to talk about now is I want to talk about ensemble analysis. So, as we've said, flexible systems sample a large number of conformations. So, ensembles of conformations, which is really just a fancy way of saying kind of a group or a set of conformations, are the most appropriate way to represent the state in solution. We can't talk about a single state, we have to talk about groups of states that it might be in. And so there are a bunch of different ensemble analysis methods, but they all use the same following approach. You start out by computationally generating a large ensemble that describes the possible conformational landscape available with the protein, all the conformations it could possibly be in. You then compute the theoretical SAXS profiles from the individual conformations, and you select a subensemble of conformations that collectively describes the experimental profile. So there's a number of different programs that do this. I'm not going to go through all of them. And they differ on all 3 points in terms of how you generate the pool, how the profiles are calculated, and how this kind of subensemble of conformations that describes the experimental profile is calculated. The one I'm going to focus on in this talk is the Ensemble Optimization Method, EOM, from the ATSAS package. So this was the original approach, published I think in something like 2007, and it remains the most widely used method. And this is the most recent paper on it, so you should take a look at that if you use it. So generally speaking, the approach is relatively straightforward. You have two programs in the ATSAS package, one called Ranch for random chain, one called GAJOE for genetic algorithm judging optimization of ensembles, and you use them in sequence. So first you take all the known structural information, the sequence of the protein, any high-resolution data about folded domains you have, contact information, symmetry information, you plug it into Ranch, and Ranch generates a large pool of random models, somewhere on the order of 10 to 100,000 different possible conformations. You then calculate all the possible scattering profiles of those combinations, and then you use this GAJOE program to use a genetic algorithm to create kind of a small optimized ensemble of conformations that the scattering profiles of which, on average, when averaged together, fit your experimental data. You do this a number of times and you come up with a kind of small ensemble of shapes in solution that fit your data collectively. So I want to talk a little bit first about the pool generation. This is in some ways the most important step of EOM besides measuring good SAXS data, because without a good starting pool, your EOM results are not going to be valid. You're going to be selecting from the wrong pool, and it's not going to tell you anything about your system. So good pool generation requires kind of realistic and adequate sampling conformational space. You have to produce feasible models, so for things like avoiding steric clashes, and if possible, you want to incorporate any high-resolution information you have about folded domains. So Ranch itself uses an algorithm that's kind of based on bond versus dihedral angle distributions represented by the C-alpha Ramachandran plot. And this allows generation of models that resemble chemically denatured or natively unfolded proteins in the regions where there is disorder. It can also incorporate high-resolution information from multiple domains and allow those domains to be either fixed or free in your model generation. And then finally, you can generate oligomers via symmetry operations if you know there's a symmetry. So this is just an example of these C-alpha Ramachandran plots, and you can see the different kind of heat maps for the native and randomly, or rather chemically denatured disordered regions that it uses to generate possible models. So I want to talk a little bit about some of the details of using Ranch. So first off, you should know that it generates flexible structures from a single sequence. So you can't do multi-chain proteins unless you can generate them through symmetry operators. If the protein consists of known folded domains, you can input one or more high-resolution structures as .pdb files, and these input domains are kept fixed. So any amino acids not in the high-resolution structures that you input are allowed to move. So if you have, for example, a PDB, a crystal structure where you've modeled in a particular conformation for a flexible loop or linker, or maybe a flexible N C-terminus, you want to delete those so that Ranch is free to move them. Additionally, any input high-resolution structures and input sequences must match exactly amino acid by amino acid. So this is often the most challenging part of getting the input right. Not only do you have to input the PDBs in sequence order, this is very important, people sometimes mess this up, or they do reverse sequence order, but if there are any gaps in your input PDB file, like a missing flexible loop, you have to split the structure into multiple PDB files around those gaps. Otherwise, Ranch is going to see different sequences between the PDB and the sequence that you input, and it's going to spit out an error. Additionally, of course, your sequences should also exactly match the sample, including any tags like a his tag in particular, or post-translational modifications. You can constrain symmetry, you can add known distance constraints or contacts if you have them, and then you can also pick between different structure types for pool generation. So this is very important. Ranch allows you to pick random coil, which is the default, and this is a C-alpha dihedral angle distribution consistent with chemically denatured proteins. Typically what you want is you want to pick native-like, which is not the default. So this is a distribution consistent with disordered proteins, natively disordered proteins, and so for flexible linkers and things like that in an otherwise folded system, this is the right approach. You can also generate compact chains, which is, again, disordered systems but forcing compact linkers. I don't recommend this. There's no reason to bias your pool in this way. You'll see this in the EOM results. Finally, the default is to generate about 10,000 models. I prefer to generate about 50,000. But first I do a test run with about 100 models to make sure the settings are correct. You want to save the generated models in this test run, open a few of them in PyMol or whatever you use, and make sure that you're generating reasonable confirmations, that everything that's flexible is moving, everything that should be rigid and fixed isn't, and that these things aren't kind of running away. Otherwise, you can waste a lot of time generating all these models and not getting good results. So that's the model generation. Now I want to talk about how we select an ensemble using this genetic algorithm. So it proceeds like this. You start out by selecting 50 sub-ensembles of structures. This is the first step of the algorithm. And this is just done randomly. So you can also, the number of structures in each sub-ensemble is dynamically selected by the by the program. Usually it's about 50 to 20. And then from these 50 sub-ensembles, you generate 100 different new sub-ensembles using two different approaches. You can either replace 20% of the structures in each sub-ensemble with structures either from your full pool of structures or the other sub-ensembles. Or you can exchange at least two structures between two randomly chosen sub-ensembles. So this is the genetic operation, these two ways of changing your sub-ensembles. So then you have these 150 sub-ensembles. You calculate the fit for each one against the data, right? So each one has 5 to 20 different structures in it. You take the calculated profile for each structure, you average it for the whole ensemble, you do that for all 150, and you fit them against the data. You select the 50 best fit sub-ensembles, and then you repeat these generating new sub-ensembles and selection steps 1,000 times. And then at the end of this repeat, you take the best fitting sub-ensemble, and that's the one that you use to fit the data. You run this genetic algorithm itself, these 5 steps, multiple times, usually 100, to generate a final sub-ensemble. And this final sub-ensemble combines all the best sub-ensembles from all 100 runs of the genetic algorithm into a single distribution, a single sub-ensemble. And from the sub-ensemble, you calculate out the scattering profile, the fit against the data, and the distribution of kind of size and volume in the sub-ensemble. And this represents the conformational space accessed by the protein in solution. So the genetic algorithm also outputs the structures of the single best fit sub-ensemble, so that's from any individual genetic algorithm run. So this is kind of a small, right, again, 5 to 20 member output, and it doesn't represent every possible state in solution. It just represents a few conformations, so you have to be very careful not to overinterpret these output structures. They can be useful for understanding what it means to have, say, a compact structure versus an extended structure, but you shouldn't present them as the states in solution. They're just a few representatives thereof. So that's how it works. I want to talk about some tips for actually using it. So one interesting thing is you can use this genetic algorithm on a pool of structures generated by Ranch, which is what we typically do, or by another program. So if you have another way of generating the structures through, say, a Monte Carlo MD method or something like that, you can then take that pool and put it into the selection algorithm. Multiple pools of structures can be used, example combining monomer and dimer pools, but this is generally not recommended. So then you're asking a lot of your selection algorithm to be able to pick out these different conformations. The other kind of trick is you want to make sure you specify a sufficient number of spherical harmonics for calculating the scattering profile of each structure. So this is using Chrysol, which uses the spherical harmonic method. For particularly compact structures, the default of 15 is fine, but if you have large extended particles or maybe really extended IDPs, you should add in more harmonics, and you can go up to a maximum of 50. A good way to test this is using that test pool of structures that you generate, that first 100, find a few that look particularly extended, calculate their profiles with Chrysol with the different number of harmonics, and see if the profile changes. The reason you don't just want to default to the largest number of harmonics possible is it's going to take a lot longer to calculate the profiles for each individual conformation. So you want to check this as well. I typically run the algorithm 10 independent times, and I compare the results to make sure that we've kind of reached a stable point in the algorithm and it's converged by the end. And I also want to note that EOM can be run as a single command, EOM, or you can run ranch and GAJOE separately from the command line. And you can rerun this algorithm, this GAJOE, with the same pool without rerunning ranch if you just want to change whatever kind of settings you're using in the algorithm itself. So that's how we run it. Now I want to talk about what the results look like. So the main EOM output is the radius of gyration, maximum dimension, and volume distribution of the selected ensemble. So instead of talking about individual profiles, what we do is we talk about kind of what does the range of shapes look like. And so what we do is we take all those shapes that we found in the genetic algorithm step, all the different conformations. For each one, we calculate the radius of gyration, the maximum dimension, the volume, and the C-alpha average distance. And we also do that for the full pool of possible conformations in solution. And then by comparing these two distributions, one for the selected ensemble, one for the pool, we can make statements kind of about the degree of flexibility of the protein relative to the range of shapes it could be adopting. And so a nice example of that is shown here in this paper published this summer. So this was a case where we were looking at a system that has a folded catalytic domain, shown here in blue, and then this intrinsically disordered region, shown here in a variety of colors. This is different structures output by EOM, but more importantly, we have the distributions output by EOM. So in blue, we have the distribution of sizes, in this case, radius gyration from our pool. So this is all the conformations that the protein could be in solution. And in red, we have the selected conformations, so the conformations that it actually is adopting in solution, the ones that fit our scattering data. And so what we're seeing here in all cases is that these selected conformations are really kind of narrowly focused down at these very small size ranges. So this is telling us that instead of adopting this kind of completely disordered shape in solution, this intrinsically disordered region is characteristically adopting compact conformations close to our catalytic domain. And you can see that in the sample structures as well. And so based on this, we can then go on and talk about kind of reasons why that might be happening and what biological information this is giving us. So this is the main result of EOM is these distributions and where our selected ensemble fits versus the pool. So EOM also provides some advanced metrics of flexibility. So there's the R-flex. So this is a quantification of flexibility of the pool, either the total pool or the selected ensemble, which allows you to directly compare between flexible and rigid systems. It's usually represented somewhere between 0 and 100, 0 being completely inflexible, completely rigid, 100 being completely randomly distributed and completely uniform, and it's a percent. And then we also have this R sigma factor. So R sigma is the ratio of the standard deviation of the selected ensemble and the pool. So if you think about it, if your selected ensemble is the same as the pool, it reproduces the initial distribution, R sigma should be 1, because the distribution of sizes in your selected ensemble is the same as the distribution of sizes in your full possible set of conformations. On the other hand, if your selected ensemble is more narrowly distributed than your pool, if R-flex of the selected ensemble is smaller than that of the pool, you expect R sigma to be smaller than 1, and this is because you have a tighter distribution, so you have a narrower standard deviation. So when you take the ratio of the selected ensemble in the pool, you end up with something less than 1. Conversely, if your system is more flexible than the pool, R-flex of your selected ensemble is larger than that of the pool, you expect a wider distribution, and so you expect R sigma greater than 1. And so if R-flex of the selected ensemble is significantly smaller than that of the pool, and R sigma is greater than 1, there might be a problem with your EOM result. And that's illustrated in these distributions over here. So here we're seeing black as the pool that was calculated, and then the one case where we're seeing this kind of extreme R-flex less than the pool, R sigma greater than 1, is this bimodal distribution shown in red. And so in this case, we have very narrow distributions. So we're getting a small R-flex, but because we have two of them, there's a large standard deviation, so we get a very large R sigma value. And so in this case, you want to go back, you want to verify that your data and your inputs are all accurate, that you're generating good models, and you need to be very cautious before you trust this kind of distribution, and these statistics can help you figure that out. I do want to note that in ATSAS 3.0.2 and previous, there was a bug in the EOM R sigma calculation. It was significantly off. They fixed this in ATSAS 3.0.3, which was released just a few days ago relative to when I'm recording this talk. So if you're using an older version of ATSAS, you should recalculate R sigma directly from the output EOM distributions, or better yet, just upgrade to this newest version of ATSAS where it should be fixed. So now I want to give just a few examples that use EOM. So the first of these is from a paper published this summer out of the Tesmer lab. This is data they collected at BioCAT. So I'm not going to go into the details of the system, but essentially they're interested in this system that has roughly two domains, this DHPH domain and then this DEP1 domain, which are connected by a flexible linker. And so we had crystal structures for both domains, and we collected SAXS data on the system with and without this DEP1 domain and this flexible linker. You can see here kind of just metrics of the SAXS data quality, which are good. And then we carried out EOM analysis on our systems. And so what I want to draw your eye to here is the distributions for the system with the flexible linker. You can see it's generally speaking adopting significantly more compact states than the possible range of conformations in solution. And so using this combined with other structural and biophysical techniques, we were able to show that it is adopting these compact states in solution, and that that's most likely attributable to an interaction between the domains, and based on this association, proposed a mechanism for inhibition. So that's kind of a fairly standard use of EOM. The second case that I want to highlight is a really kind of advanced use of EOM. So this comes from the Pollock lab at Cornell, which, among other things, studies nucleosome dynamics. And so they were particularly interested in DNA unwinding from the histone core, which is essential for transcription, replication, and repair. So what they did is they came to BioCAT, and they carried out time-resolved, contrast-matched SAXS experiments, where they blanked out the scattering from the histone core, and they could look at just the DNA, and they studied the dynamics of this DNA unwinding with SAXS. And so they measured a range of time points, which you can see in this figure A here, and each measured time point contains essentially a continuous distribution of different unwinding states. And so they had to fit each state using EOM to figure out what was going on in solution. So this was a challenging system. You weren't able to generate candidate structures using Ranch, so they used custom methods. And then they carried out the ensemble selection using GAJOE at each time point. And so we can see the results here. So what we're seeing in part A of this figure is the RG distributions from EOM. So you can see at the earliest time point we can measure, there's a distribution, there's some in this compact state, some in these intermediate states, and a little bit in the extended state. And then as you go out to later and later times, you're shifting the amount from these compact states into these intermediate states, and then eventually into these fully extended states. And so using the models that they created and the EOM selection, they were able to give some examples of what the wrap state, the extended state, and the different intermediate states might look like. So you can get an idea of what's going on at each different size. And then with some additional SAXS data from equilibrium conditions and from some FRET data that we used to verify the results, they're able to put together a really nice kinetic picture of the DNA states during unwinding. This is a really challenging and a really, I think, fascinating and powerful application of EOM to SAXS, and I recommend you take a look at this paper if you want to learn more. So just to summarize then, SAXS is a really powerful tool for studying flexible systems in solution. It's one of the few methods that can quantitatively characterize partially disordered or completely disordered macromolecules. You should use multiple indicators to determine whether a system is flexible. Other effects like aggregation or extended overall shape can show the same effects on the profile and parameters as flexibility, which is why you need these multiple indications. Use ensemble analysis to inform about the overall set of conformations sampled in solution by your system. Rarely, if ever, is it appropriate to discuss a single state of the flexible system in solution. And then finally, as far as ensemble analysis goes, EOM in the ATSAS package is the most common tool. but many others are available. So with that, I'm just going to leave you with some references. These are great papers to check out if you're interested in flexibility in SAXS, and I thank you for your attention.